This is episode 126 of the Immunology Podcast, Functional Oncogenomics with Dr. Daniel Peeper. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoyed the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Daniel Peeper from the Netherlands Cancer Institute, Brenda's old home, on the podcast to talk about his research using powerful functional-based genomics to screen for novel therapeutic targets to tackle tumor heterogeneity, immune dysfunction, and therapeutic resistance in cancer therapies. We've also got our usual roundup of recent highlights in immunology news coming up, but first...
The American Association of Immunologists Immunology 2026 program is shaping up to be an exciting and dynamic one. From cutting-edge research and scientific sessions to can't-miss lectures and networking events, this year's annual meeting offers something for every immunologist. Join AAI for its 109th leading all-immunology conference in Boston this April 15th through the 19th. Explore the amazing program and register today.
You ready to park the car in Harvard Yard?
Yeah, I'm excited. I've been to Boston once, but I didn't visit a lot. So where are we going? Where are you taking me, Jason? You're almost like a local there. So what are the non-miss places?
So, the number one I think I need to do is take you on the Mass Pike, which is a road, and let you see God flash before your eyes as you drive through Boston combat driving.
What?
Boston drivers are notorious. Like, they will pass you on the shoulder of roads at 90 miles an hour. And so if you want to have a religious experience where you see your life flash before your eyes, you just go on the freeway during rush hour. That's great. So we should do that.
Not.
You could do duck boats. That's classic.
Okay. Duck boats.
Yeah. So they're like a boat. They're like a car boat that could go into the water in the Charles.
Into the water. It's like a hybrid
car boat. Yeah.
A car boat. Okay, now I understand. Okay.
If you really like American Revolutionary War history, you can see where half the stuff started.
Okay.
If you care about that, or you don't.
Oh, the Boston Tea Party.
You can see where we're going to be right at the seaport, so you can see where the Tea Party was. You could do historical tour.
Yeah.
You could see the town halls and stuff. Harvard's from the 1600s, so it's not anything compared to Europe. But if you want to see some of the original American buildings that are still there, there's all of that. So it's the most historic of the, I would say, of the kind of American experience. It doesn't get much older than Boston.
But enough history. This is not where our listeners are tuning in.
But you want to know what I'm going to go deep into?
What?
Sequencing. Epstein-Barr virus.
Wow. What a segue. Okay. Another very old thing.
Yeah, very common thing. Like the paper cites 90 to 95% of the global population has EBV.
I know, right?
Yeah.
I wonder if in 100 years when we get vaccinated for EBV, people are going to think that we were barbarians for just allowing ourselves to have this virus. Just we have to live with it. Like people live with tuberculosis before or something.
Yeah, that's a good question. Do we all devolve? Maybe we all lose our superpowers.
If we stop being infected with EBV, you say?
Yeah. Like something super fundamental. Like we can't speak anymore because it's really in charge of our language. Okay, we're going wild with it. All right.
Yes. Okay. Focus, please. So, sequencing.
Been a week. All right.
So, dear listeners, it's been a long week. So, please understand. All right.
All right. It's in Nature. Host control of persistent Epstein-Barr virus infection. It was accepted on 12th of February, and it doesn't have a publication date because it's accelerated. It's an accelerated article preview. So take that. We're super fast here. First author is Axel Schmidt. Last author is Kirsten Ludwig. And it doesn't have any of the disclaimers yet of other co-firsts, so I apologize. It's a preprint. All right. So high level. EBV's everywhere, right? But, most people just have latent infection that's hiding in your B cells. And they wanted to see how much of it was actually popping out and being transcriptally active. And it's really hard to do. Like measuring antibodies, it's a pain. It's very hard to do. So they took a UK database, biobank, and US biobanks, and tried to see if blood-based genome sequencing could detect EBV. And it could in about 20% of people. depending on the data set. You could get short pair read pairs mapping the EBV genome, popping out. Usually most of the time it was like one read. But you absolutely can detect like very low active, like almost non-existent, but like it's just a little bit there, EBV in 20% of people. Like, so it's popping out.
But it's like integrated. It's like the integrated EBV.
Well, but they're showing that when they do the sequencing, that you're seeing it appear, right? Like, so you're seeing it come out, and right. Remember, so you when you even do sequencing, you don't always get literally everything. You get more of the active, right? It's not RNA-seq, it's DNA, right? But like you're still going to have a bias to, it's like, oh, it's not integrated or not integrated. Is it detectable, right? And so you're seeing, and so this population, especially the higher amounts, because they say, oh, then there's some people with even more reads than one, right? And what makes them? Well, if you have active HIV going on, you're a more high read. If you have MS or certain alleles in MHC class 1 associated with MS, or MHC class 2 associated with rheumatoid arthritis, remember diseases we've talked about before that are associated with EBV, you see more reads in those people. So it's correlating back. And then they saw polygenetic overlap with IBD, hypothyroidism, and type 1 diabetes. And they then found other HLA phenotyping that mapped with it, as well as resistance genes that were found that were already known to be resistant, mapped with less. Right. And then higher viral loads of EBV based on these counts was associated with things like immunosuppressive drug intake, HIV, smoking, things you'd expect. So the point being, it's really one major experiment with all this orthogonal analysis, right? You can detect EBV viral load surrogate as a surrogate to viral load, which is very hard to do with genome mapping. Like, oh, we can see it on the genome read, because, you know, you don't get everything every time you measure it. And something rare like this is hard to see, but you can see it. And then when you see it, yep.
But then you said just 20%. So if we say 95% of people are infected, but then you only get reaching 20%.
Correct, because most people are so latent you don't see it.
All right.
And then they're seeing, and then a lot of people only had one copy, and then people with 2 and then 3. The more copies you had that were detected in a person, the more likely you were to have HIV, active HIV, or be on immunosuppression, or smoking, or have other associated genetics known to go with like MS. And MHC class 1s that are associated with MS, like HLA-A02:01, right? Or MHC class 2 is associated with rheumatoid arthritis, or all these other things we know track with EBV and EBV-associated diseases. So they're saying if you see more of it, then you have this. There's also an MHC that I'm not going to dig through to find the exact one here, but there's an MHC that's less likely to be susceptible, and a couple other genes that are EBV resistance, and those were decreased in people with high copy numbers. And so they're saying, because there's says hundreds of thousands of people in each bank, 480 in one and 336,000 in another. And so they're seeing, hey, the things we know track with EBV genetically or as other markers also track here.
Right. Okay.
And so they're really saying you could do this as a surrogate for understanding EB infection.
It would make sense that you would follow those patients that have this, you can detect the EBV.
You might, or if you're trying to figure out how to screen or understand the disease in populations, the viral load for EBV is very hard to do. Is it an active infection? It's not. If I have IgM, how long ago was I really infected? Did I just have a B cell pop out of into activity for a day or two, could trigger an IgM response and go away, and it's hard to measure. But then sequencing now is getting cheaper and cheaper, and so it can kind of be more of a shotgun approach that's less immunological, but gets at this immune marker of EBV status maybe more efficiently in the end, which is not intuitive, right? Like sequencing, like, why would I do that? But the point being, you could do that, or you could target sequence for just these genes, right? Instead of the whole thing, but you can know that on a population level, you could detect it and detect meaningful differences now, which they didn't know before. So from someone who is concerned with EBV in general, because it's one of those things you care about in donor populations and screening, it's not really with what I do, but it's like a general thing that EBV, what the heck do you do about EBV, right? Even 90% of people have it, but like, what do you how do you screen organ donors for it, for instance? Like,
right.
You can't, but you want to. This is cool.
Yeah. Okay. That seems like a useful thing to have.
Yeah, it was super good. It's cool to know. I'm like, that works. Wow.
All right. Well, thanks for sharing. For my first paper of today, do I have a segue? Well, my segue, so it talks about B cells and EBV affects B cells preferentially. So
That's
that's a
less
segue.
bad than mine.
Is that an acceptable segue?
Yeah.
All right. So the paper I'm going to talk about is called Lymphocyte-Derived Cholinergic Circuits Modulate Germinal Center Output and B Cell Activation. And was published in Nature Immunology on 24th of February. First authors, Duygu Nekanitsky, Logan Smith, and Robert Nekanitsky. Yes, that's the last name. So shared last names. I wonder what's the story behind that. For corresponding author, Tak Mak. And this is at the Ontario Cancer Institute in Canada. And in a way, I think, again, these titles are very descriptive, but we know, so it's cool because it's, again, one of these papers in which they're looking at something like a compound or a kind of physiological circuit that is normal in a non-immunological context. And then he looks at, is it relevant for immune cells? And in this case, is about acetylcholine signaling, which acetylcholine is a neurotransmitter and very important for the parasympathetic nervous system, and excites. So basically, it excites muscles by contraction, modulates brain function. So it has a lot of kind of very neuronal effects, and is very well known for the neurologists in our listeners will probably be very familiar with this. But it turns out that there is also a role for acetylcholine on immune cells. We know that immune cells, it has been, I would say, fairly recently shown that immune cells express, or that probably was known that they express the receptors, but these receptors actually do things like the sensing of acetylcholine by immune cells can affect their function. And in this paper, they are looking into the role of both acetylcholine production and sensing in lymphocytes during the selection processes that happen during germinal center formation and maturation of B cell affinities. So basically what they do in this paper is that they are, they start by asking the question is acetylcholine being produced by T cells or B cells or rice cells during a normal germinal center formation? So they do some, classic experiments for studying germinal centers in which they immunize mice with these sheep red blood cells, and this is a very well-established protocol. And they show that indeed, if they have this GFP reporter for the enzyme, which is choline acetyltransferase, which is the one that makes the neurotransmitter, that they can see that this enzyme gets activated in both B cells and T cells in the spleen of mice that are undergoing this immunization. And they have these mouse models, the flux mice, in which they can delete the acetylcholine transferase in either T cells or B cells. And they start to kind of understand what happens to the immune response when they do this. And what is interesting is that they show that there seems to be a function for this acetylcholine production by both T cells and B cells, because the absence of this enzyme affects the quality of the affinities and the clonality of the B cells that come out at the end stage of the germinal center. So they show that when they have the loss of chat, I'm going to call it, the choline acetyltransferase, results in more low affinity clones, so kind of in a reduced overall presence of high affinity clones. And they show this because it's a cool system in which they have a model antigen, and they have a specific B cell clone that recognizes a model antigen. And then there's a second antigen that is recognized with a lower affinity. So the first antigen will give a very strong activation of the B cells, and this transgenic B cells would have a very high, they don't have, so these B cells don't have any absence of acetylcholine, but then when you compare them with the endogenous response, having this high affinity or middle affinity response that you can track, it gives you kind of a way of evaluating how is the endogenous response in the same mouse. And what they see is when they compare the resulting endogenous response to this would be this baseline or these kind of marks that that are these transgenic B cells. They show that absence of acetylcholine results in endogenous cells that have lower affinity. And the conclusion is that the loss of chat allows these lower affinity clones to not be outcompeted by the high affinity clones that they are putting transgenically. And therefore, there's this idea that there is certain negative feedback or kind of a negative limitation on the strength of the signaling that these B cells receive, so that only the very high affinity clones are the ones that receive the stimulation and the survival signals, and those are the ones that are preferentially generated during the germinal center response. So they do. They also analyze. So they kind of with this in mind, they compare RNA-seq in both, and so they show that T cells and B cells affect this response in different ways. And so they look into how are the B cells or the T cells with or without chat, how are they looking in single RNA-seq, and they use this GFP reporters. And they show that the B cells that are expressing chat, they have kind of a more activated phenotype. And also the T cells, the follicular helper cells that are expressing acetylcholine have increased of T follicular helper markers, indicating that this are driving this B cell helper, that these follicular helper cells that are driving the B cell help, they are also the ones that are expressing acetylcholine. And they show that there's the way that this connects is through affecting the signaling downstream of the B cell receptor in the B cells, and that's how it kind of modulates the response of the B cells to the antigen stimulation, and that by having this acetylcholine signaling that is made by the follicular helper cells and is sensed by the B cells, that is how it's reducing BCR signaling and therefore increasing the competition and the selectiveness of the stimulation that the B cells receive during germinal centers. So again, I think it's really cool because you have these situations in which you have something that has always been, thought of just like a neurotransmitter. It has a sense that seems to have a very specific effect on immune responses. So I think that's really cool to see this kinds of studies.
So you know how Benadryl makes you sleepy?
Uh-huh.
That's because it's anticholinergic.
Okay.
So what does that mean? So if you're turning that off, then that's going to do what to your immune system if you're taking it to sleep?
Well, if you get vaccinated at the same time, if you take
what you do for your allergies, right? You often will take Benadryl on top of a vaccine to prevent your arm from swelling up.
Will you? Okay.
Sometimes people will. Yeah.
Well, I guess in this situation, if you are inhibiting, I guess you would have to do it many times because, of course, the germinal centers start forming like 5 days. Like the peak is like 5-7 days after. So I guess that if you're interfering with that mechanism, that acetylcholine signaling, then you are going to allow lower affinity clones to survive the germinal center formation in detriment of the high affinity clones, which is probably something you don't necessarily want.
Yeah. but interesting.
Yeah.
Complicated.
I know. Everything is immunology. In the end, everything, just all the roads lead to immunology.
True. All right. Well, you know what else is complicated for my segue? HIV.
Definitely.
All right. In this one, also an accelerated article preview accepted the 19th of February in Nature. It's dynamic antigen expression and cytotoxic T cell resistance in HIV reservoir clones. Let's see here. Only one first author, I think. Isabella Ferreira is the first author, and Brad Jones is the last author. All right, so here we go. Oh, wait. We may have, nope, two first authors. Got a second one. Alberto Herrera found it. All right. So we know HIV is a difficult bugger, right? Because even if you suppress with antiretroviral therapy, there's these reserve cells that stay around. And as soon as you go off, out pops HIV. Also, HIV indiscriminately is relatively an indiscriminate retrovirus. It kind of goes anywhere it can. It's not like, well, there's one insertion site for it. And so there's been a lot of work about how could I do a protocol. I have you on antiretroviral therapy, but I can't clear these last bugs. Is there something I could do to make these last infected cells like be killed by the immune system? And so there's this idea of kind of activate them, have them appear, pump out some, do something, and then get them killed. But they're very hard to study, as you can imagine. So this group, and I think the reason it's a Nature paper is they're actually able to play with authentic reservoir clones, as they call it, these ARCs, that proliferate and accumulate while producing infective virus. But these ARCs do not succumb to cytopathicity, so they don't die to cytotoxic T cells. So they first are just able to get some and characterize it from HIV patients. And that's pretty hard to do, right? Just getting these cells and being able to do anything on them was a challenge. And so then when they get them, they show that if you co-culture them with CD8 cytotoxic lymph T cells, they cold proliferating, generally speaking, but there was a subset. So if you sustain them, if you just keep them in culture for a long time with a lymphocyte clone, it works and you can kill them. But the ex vivo T cell response was not great, and it only occurred slowly. So if I just take a super clonal expanded T cell, you get killing. So obviously the host's blood cells aren't working. Then they really target it and find that there is a Treg ARC, right? So a Treg T cell that has HIV in it, right? And these really don't die to cytotoxicity. But, and then they're trying to understand how they get killed. And these ones are particularly resistant because they have low oxidative stress. So if you give them deferoxamine, which is a oxidative stress inducer, then they'll start to die. An epoxy inducer, and it's an FDA approved drug for other stuff. So they're really, and then they spend a bunch of time just trying to ferret out, well, can anything else kill them? And then they look at different clones. And so they find that depending on where the virus integrates into different regions, or, the virus prefers zinc finger binding regions, and those tend to be more active and more killable because they're just more actively producing virus, which then makes the body go, oh, you're being bad. But then some other spots where it integrates, like you don't get a lot of activity, but it also doesn't want to go away. But the real culprit for this reservoir that they found that you can't kill it are these Treg reservoir cells. And that they just don't act like, regular T cells are pretty, have high oxygen stress because they're going to kill other things. And these don't. And so you have to turn oxygen stress on in order to kill them. But yeah, the Tregs are a problem.
They're zombie Tregs.
Well, they're not dead. Right.
Well,
they're just harbingers. Yeah. They're just pumping out HIV for you once in a while.
Huh. That's right. I mean, it's interesting because a very clear what's the word? consequence of the metabolic differences and between Tregs and conventional cells. And it's funny that it results in this. I'm surprised, and I feel a bit I'm a bit embarrassed for these Tregs that are playing against the host, but we know they do it sometimes in cancer and now apparently also in HIV.
Tisk.
It's interesting because I wonder if you could, you can't really deplete Tregs because we know what happens when you do that. But it would make sense to try to target Tregs in some other way. All right. Very cool. Very cool.
Yep. All right.
So last discussion of the day. I have a fairly clinical paper now, and I really like. I think I like it again because it's another paper looking into novel therapies, immunotherapies for autoimmune disorders. And I think that, as a consequence of all the CAR T cell for autoimmunity, there has been such a revival in finding ways of helping people that have these severe autoimmune diseases that are mostly mediated or characterized by these high titers of cell autoantibodies. And after the CAR T cell, the great success that CAR T cells have had for this, people have been trying to find other ways of exploiting the same principle, but maybe, making therapies a bit more accessible because CAR T cells are still expensive. It's still a lot of work to make them. And so this paper is called Bispecific T-cell Engagers for the Treatment Refractory Autoimmune Connective Tissue Diseases. And it was published in Nature Medicine, 19th of February. First joint authors, Christina Dusing, Andrea Ermina Giorfri, and Ayla Nadia Stutz. From senior authors, Wolfgang Merkt and Jörg Distler from the University of Düsseldorf and the Fraunhofer Institute. And in this is basically a report from a clinical study in which they basically do a compassionate use clinical study of 10 patients that have this severe autoimmune connective tissue diseases that have been, they're not responding to regular therapy, corticosteroids, cyclosporin, things like that, killing the immune system basically. And they're also refractory to single kind of rituximab therapy, so anti-CD20 therapy, just like alone. So what they do is they test two engagers, T-cell engagers, called, and I have blinatumomab and teclistamab, which are bispecific engagers that have, on the one hand, an anti-CD19 for blinatumomab or anti-BCMA for teclistamab, and on the other, next to it, is an anti-CD3. So basically what you do is this molecule is designed to bring together a T cell and a target cell and induce kind of killing. And these therapies have been originally developed for blood cancers. Usually blinatumomab is used for acute lymphoblastic leukemia, ALL, and teclistamab is used for multiple myeloma. Now, these people, they decide to repurpose for these diseases. And we have two diseases, anti-synthetase syndrome, ASyS, and systemic sclerosis. Basically, you have antibodies that are targeting autoantigens, transfer RNA synthetases on one hand, and antibodies, again, I think most of the time topoisomerase, fibrillar antibodies. And in the second case, I don't think it's important what so much, but usually you have inflammation of the muscle or the connective tissue, and it can be very severe. So you have like massive organ involvement because you have connective tissue all over the place. So what they do is that they have these 10 patients and they treat them. They have like a combination therapy. And so I like this paper because this is it. This is the kind of things that, they come from this is the kind of clinical trials that start with, hey, let's see what happens when we treat patients like this. And I think it's very nice because it was in general a fairly successful trial. So they treat the patients with either they have anti-CD19 engager for ACEs because that had shown some already some success with anti-CD19 therapies, and so they think that would have been a good choice. What are you going to treat the patients with? They thought that CD19 targeting was the best for this. But then for systemic sclerosis, they are targeted BCMA, which is this molecule expressed on plasma cells, which are the ones that are making the antibodies, and so which is not the case for CD19. And they treat the patients with this. They have they test out different regimens as patients. They have 5 patients for each treatment, and they test. But basically, they give some length of treatment depending on how long the antibodies last in the patients, and then they maintain the patients with rituximab, with anti-CD20. So it's like first you have a hit with anti-CD19 or anti-BCMA T-cell targeting, so bispecific engagers, and then you maintain with rituximab, which most of the patients had already shown to be refractory to just rituximab. They would, reduce B cells, but then the B cells will come back. And basically what they see is they see really good results in basically all of the patients. They see improvement in their inflammation. They see improvement in their clinical measures. So when they look at the first patients, the patients were treated with anti-CD19, the ACEs patients, basically they have a lot of muscle inflammation. They cannot walk. They are bedridden, and many of them, improve a lot. And they reduce all these markers of muscle damage. They're reduced, and they really look a lot better. So some of the patients were followed for a year. Some of the patients were followed for a couple of months. So there's some time amplitude of the follow-up at the moment that the paper was written. And what's interesting is that for most of the patients, they see a reduction in antibodies. For example, this is anti-Jo1 is one of the most common antibodies that is characterizing this disease, and it's believed to be a mediator of at least some of the clinical symptoms. And in most of the cases, they see a reduction in the titers, but not a complete elimination. But you still see clinical effects and clinical advantage. And similarly, these patients, they still keep enough titers of protective antibodies against, I think they measure rubella or measles or normal antibodies that are a consequence of vaccination, for example. And in most cases, they keep anti-measles, anti-mumps, anti-rubella. They keep those IgGs that are still present in these patients. And that's very interesting because that means that most of the cases, these patients don't require, for example, IVIG treatment, which is undesirable because that means that you don't have antibodies to protect you. So most of the patients with ACEs treated with CD19 engagers, they get better clinically. And although they reduce their general antibody titers, it's not below the lowest acceptable levels. The story is different for the anti-BCMA engagers. In all cases, they look at muscle biopsies, and they show that there's a lot less infiltration of B cells, but also T cells, and in general, less inflammation. CD4, CD8, the numbers, and the MHC class, the MHC expression on these tissues is reduced substantially for both treatments. And for the systemic sclerosis, in which they get this BCMA that is better at targeting plasma cells, they show that these people do get a very strong reduction in the titer of the antibodies that are come with this disease, but also antibodies that are meant to be protective. So some of these patients, they have to undergo IVIG treatment to compensate for the lack of B cells making protective antibodies. So in all cases, B cells are gone. They have very, like CD19 positive B cells are very much reduced and in blood and in the biopsies. But then you still have, which in a way, it explains a lot of the improved symptoms and improved condition of the patients. It shows that it's not only about not having the antibodies, which in the first case, they still have the antibodies, but the B cells themselves are big drivers of inflammation, and getting rid of those seems to suffice to keep the patients at least healthy, and they will probably need this treatment in the long term, but they, for example, don't need to take most of them don't have to take corticosteroids anymore, which is really good. So again, I think this is another really nice example of how this new line of therapy is giving really cool results. And I'm looking at some of the details of the patients. They are people that some people have had these diseases for years. There's one of the patients had systemic sclerosis for 18 years. One of the patients with ACEs, she had breast cancer, hepatitis, and on top of that, she had this. Some people had such severe damage in the tissue. They had amputations. I can imagine what it must have been to be in the doctors treating these patients, and the patients themselves. They were running out of options, and this all of a sudden seemed to. There were some adverse effects. Some people had to be treated with some corticosteroids to reduce it, but in general, all of these patients benefited from this treatment. So I thought it was really cool. I really liked seeing this. Sometimes it gets a bit boring because they're basically just describing patient by patient what happened, but it's so exciting to see this kind of studies being done.
I mean, I'm a big fan of bispecifics. I think they're super cool. I think there's probably more and more they can do, and they're technically challenging, but very awesome.
Yeah. It was so cool. So cool to see. So I'm. Let's see what other diseases are. Now everybody's going, every autoimmune disease that has a B cell, people are just trying, not just throwing these things and seeing what happens. And I think so far, I mean, many of these results are very encouraging.
Promising, I agree. All right. Well, what's also promising is this awesome conversation we have coming up here. But before we get to that, performing multiple rounds of cell isolations can be inefficient. Using the new easy-to-use 250 EZ-SEP magnet, you can scale up your cell isolation and process large volume samples like leukoplaques and whole blood in a single round of separation. Obtain highly purified cells from samples of up to 225 mil in a single step. Learn more at www.stemcell.com/easy250magnet.
Now for the second part of our show today for our interview, I'm very happy to have with us Professor Daniel Peeper. He's a professor in functional oncogenomics at the VU University in Amsterdam and group leader at the Netherlands Cancer Institute in Amsterdam. The Peeper Lab uses function-based genomics to screen for novel therapeutics and to target novel pathways to offer solutions to tumor heterogeneity, immune dysfunction, and therapy resistance, which are some of the most important challenges that now, especially current immunotherapies, are facing to reach more patients and really become more effective. So I'm very happy to be talking to you, Daniel, today. Welcome to the Immunology Podcast.
Hi, Brenda and Jason. Thank you for inviting me.
Thank you for being here. All right, Brenda. Apparently you know him, so I'm going to let you start. I'm outing you that you know him.
Yeah, so I did my first postdoc at the Netherlands Cancer Institute, and Daniel was part of his group was part of the department. So I had the pleasure of spending a lot of time with his group, and I'm aware of Daniel's work. So I'm very excited to be talking to him today here in the podcast. So Daniel, why don't we get it started? Help our listeners understand. You've been at the Netherlands Cancer Institute for a long time, so you've been working on cancer for a long time. I know that your research interests have kind of shifted and have adapted with time. So why don't you give our listeners a little brief introduction to who you are and how you got to where you're working on nowadays in your lab, helping immunotherapy get better, helping patients that don't respond to current treatments, and finding novel targets to improve therapeutic options for different patients.
Yes, this would be my pleasure. I trained as a molecular cancer biologist and got my PhD in Leiden University. Then moved on to Dana-Farber Cancer Institute in Boston to do a postdoc, and I worked in the lab of a junior of the late David Livingston. And then I moved to Netherlands Cancer Institute, or NKI in short, to do a postdoc in the lab of René Bernards. And I was one of the people who developed, I think, the first generation of function-based genetic screens. And we may talk about that in more detail later. And I established my lab in 2002. And I think I had like 3 blocks of around 8 years where the focus of the lab shifted and evolved in rather spontaneous ways, driven by both my interest and also by exciting developments in the field. So the first 8 years covered oncogene-induced senescence as a tumor suppressive mechanisms. We were one of the first groups to show evidence of senescence in vivo. And there was evidence in melanocytic nevi or moles. So this work on senescence brought us into the melanoma field, which also was very exciting at the time. In 2002, the BRAF mutation was discovered as a frequent event in melanoma. And that was soon followed by the introduction of BRAF inhibitors. And patients responded initially really well to these inhibitors, but then almost inevitably, after half a year or so, most patients would develop resistance. And so the second block of 8 years, we studied these resistance mechanisms, tried to identify novel therapeutic targets, also using screens. And then that's, I think, an interesting anecdote. I attended a melanoma conference in Zurich, Switzerland, where they reported the first results of combination immune checkpoint blockade in melanoma. And when they showed some of these water plots with the bars going down, there was a spontaneous round of applause. And I had never experienced that before, and actually never since. And everybody understood this is a watershed moment. And I shared that excitement. But I also realized, having worked on resistance to BRAF inhibitors and other precision medicine targeting the tumor cells, that melanomas are so flexible, so adaptable. They have so many mutations that I was expecting that also for immunotherapy, this would develop as a major problem, so resistance. And so after this conference, I went to the lab, back to the lab, and I said, guys, I think we should start working on a problem that doesn't exist yet, namely immunotherapy resistance. And for me, that was a big leap. At the time, as you know, Brenda, I hardly knew the difference between a B cell and a T cell. So it was quite a big investment. I had to read Janeways. I went to immunology conferences. I recruited immunologists. But, in general, I think it's a healthy step for a PI to switch topics every now and then. And looking back, it made sense because immunotherapy resistance has become a major problem.
Yeah, that was some forward thinking. I can imagine, right? Being there at that moment, looking at those early results from immunotherapy, must have been really cool to witness that, because it's such was completely new way of treating cancer, and especially melanoma that responds so well compared to previous therapy. But I guess that indeed the current problem is that it works for 50% of people, and then the other 50, something is not working. And maybe you can talk to us a little bit. How do we find ways? So we know that immunotherapy in theory has to work, right? You're activating the T cells, they are the killers. So where do we think the weak points are? And where are our opportunities to look into ways of targeting these weak points, or reinforcing, or going around them? So how are you in your lab going about this question?
Yeah, that's a great question, Brenda. So I guess summarize the lab interest by saying that we're interested to translate what I call the cancer-immune dialogue. So as you said, immunotherapy has been unprecedented in achieving clinical benefit, but it is still limited to a small number of cancer patients. And we think that if we really want to increase immunotherapy impact, we first need to be able to better translate this dialogue between the cancer and immune system. And I use the word translate in two senses, literally and figuratively. So we really want to translate the language, and how do the tumor cells communicate to the immune cells, and make sense of that communication. And once we have a better mechanistic understanding, we can translate those insights into therapeutic strategies to increase immunotherapy impact, to prevent or overcome resistance. So the lab is focusing on three major questions, and this is how we try to better understand those mechanisms and come up with solutions. So the first question we ask is what the mechanism is of tumor cell intrinsic resistance to immune cell killing, and specifically T cell killing or NK cell killing. The second question is, what are the mechanisms that drive T cell dysfunction, which is also a very big problem in immunotherapy. It's the basis for checkpoint blockade antibodies, but as you know, this can reinvigorate only part of that problem, solve part of the problem by reinvigorating T cells. And the third question in the lab is how we can better understand these functional interactions at the cancer T cell interface. And to study this, we take what I would view as a reductionist approach. We take defined cell culture models where we allow the tumor cells to communicate with the T cells. So we have paired antigen and TCRs, we have the right HLA, so there's communication. It's a standardized system that we can plug in really in any different tumor type. And when we have a cell panel with all these interacting tumor cells and T cells, we see that some tumors are highly sensitive to T cell killing, whereas others are highly resistant. And that creates an ideal setting for function-based genomic screens, where we can try to break the tumor intrinsic resistance, or we can take sensitive tumor cells and make them more resistant. And in this way, we walk from both ends of the spectrum and identify the critical players that determine sensitivity to T cell killing.
So you said functional genomic screen, right?
Yes.
What is that? Why? How is that different than a genomic screen?
Yeah, that's an excellent question. So what I like about these screens, and as I said, we've been doing them since I was a postdoc, is that they go beyond descriptive analysis. They go beyond making correlations. Description is important, making correlations is important. But what genetic screens do, function-based genetic screens, is that they establish causality. And that is very important because that sheds light on mechanisms. On top of that, we can do this now at a genome-wide scale, so we can literally interrogate the function of every single gene in the genome in any setting that we're interested in. For example, what makes T cells dysfunctional, or how do we overcome it? What makes tumors resistant to T cell killing? And with the introduction of CRISPR, This has become a remarkably powerful tool. It's clean, it's reproducible. We get very high depth. Sometimes we can, I think on average we can reproduce like 80 out of the 100 hits in the primary screen, which is remarkable. Certainly, if you compare that to older times where we used short hairpin RNAs, where this was not as efficient. And this robust tool allows us to identify pathways and indeed signaling networks rather than single genes. So these screens are very powerful.
I wanted to poke a little deeper. How? Technically. So you're saying functional genetics. How is it establishing causality? What are you doing different for someone who hasn't read all your papers, right?
So I'll try to explain it. For example, if you want to know how tumors respond to T-cell killing, are they sensitive, are they resistant, and we want to manipulate this, we introduce a genome-wide library, with CRISPR knockout library. Nowadays, we can also use activation library that activates genes. But in this example, I'll restrict to knockout screens. So every single gene in the genome is targeted by a guide that results in the knockout of that particular gene. We introduce the library into cancer cells. We treat them with T cells, and after a week or so, we harvest the surviving tumor cells, we isolate the genomic DNA, we perform PCR and next-gen sequencing, and then we look at the guide distribution. And what you typically see is that there's a dark cloud of guides in the middle. It's not enriched, it's not depleted, because most of the genes do not affect the sensitivity of tumor cells to T-cell killing. But on the enriched side, we see guides that inactivate genes that for example, affect the antigen presentation machinery. So if you make that machinery deficient, then the tumor cells are no longer able to present their antigens and they stay under the T-cell radar. And that's why those tumor cells are enriched. On the other end, you see guides that are depleted. In other words, they sensitize tumor cells to T-cell killing, and that's where they are depleted from the population. So one screen, you get mechanistic insight, both in genes that determine resistance to T-cell killing and genes whose inactivation sensitizes to T-cell killing. And of course, the second category is interesting from the translational point of view. And we have several examples from the lab where we identify genes whose inactivation strongly sensitizes to T-cell killing, both in vitro and in tumor-bearing mice. And for some of these examples, for some of these genes, we were so impressed by the effect size also in vitro, where we see complete regression of tumors, that we established a startup company to develop inhibitors to that particular gene. We're clearly not the only ones in the field doing this. There's wonderful work by Heining, Restifo, Bukervenig, Moffat, Mancuso. And now with all these efforts, we can, fill in the puzzle pieces and really build a framework of the critical determinants of tumor sensitivity to immune killing.
So maybe this is a good way to talk about. So when we think, if you don't have a lot of experience on immunotherapy, you think, well, T-cell finds tumor cell, T-cell kills. Given from your results and from maybe other people in the field, what are these other pathways that are complementary to the TCR antigen presentation pathway that are amenable for modulation one way or the other?
Yeah, that's a great question. So I think there are at least four major categories. The first category is the components of the antigen presentation machinery that I already mentioned, right? If there are deficiencies there, the tumor cells stay under the T-cell radar, they're not recognized. The second category is interferon gamma signaling, and we got a lot of information from genetic screens in the field on interferon gamma S, and we get a complex picture because I think there's a dark side and a bright side to interferon gamma in this context. On the one hand, it stimulates immunogenicity by inducing several genes like MHC, which is great for T cell recognition and elimination. But at the same time, it induces tumor T cell suppressive mechanisms like PD-L1 and IDO, and indeed by MHC induction, you can repress NK cell activity. So interferon gamma is important, but from these screens, we've seen that if you do the screen in vitro, you get different hits than if you do the screens in vivo. And that is because of the major influence of components and cell types in the tumor microenvironment on the outcome of making perturbations in the interferon gamma signaling pathway. So the third major category is TNF signaling, and my lab has focused on that pathway because it turns out that it is probably a bit more predictable than making perturbations in the interferon gamma signaling pathway. Many of the hits that are identified in screens by several different labs, including mine, that are found in vitro do reproduce in vivo. And that's genes like TREF2, RNF31, they show major effect sizes upon their activation, upon their inactivation, upon their depletion. Tumor cells do become much more sensitive to T cell killing, also in mice. And there's a fourth group, autophagy genes, where the picture is a bit less clear. There are some key autophagy genes that can determine sensitivity to T cell killing. But I think most of the information is on interferon gamma signaling and TNF signaling.
So what do you think is going to be the first drug that potentiates immunotherapy? Or what is it going to do?
Well, I strongly believe in the concept of rational combination therapy. I think it is very hard for a single agent therapy to provide cures, certainly in high mutation burden tumors like melanoma, like smoking related lung cancer. So I think that the rational combinations need to be developed. What we've seen over the past 5 to 10 years is that checkpoint blockade, in particular PD-1 antibodies, are being combined with almost every single drug that has been used in the past half century, which I wouldn't necessarily call rational combination. But it is clear that we can take advantage of the mechanistic insight that we've been building up thanks to these genetic screens and many other approaches, where we can take a rational approach, for example, by taking a drug that inhibits a tumor cell protein so that the tumor is sensitized to T cell killing, and at the same time use a drug that boosts T cell activity, which could be anti-PD-1. It has been a wonderful treatment, of course, and I think we've seen many examples of more rational approaches where you take a two-pronged approach where one treatment targets the tumor compartment and the other one the T cell compartment.
I guess one of the big risks is, well, you mentioned for gamma pathways, for example, things like interferon gamma are very toxic. So it must be, you have to find ways of combining this in which they're not excessively effective or excessively toxic. So,
but it is a bit more difficult to predict what the outcome is going to be of specific interferon gamma perturbations, because you don't know whether you're activating the dark side or the bright side, as I just mentioned. And even if you would know, it's difficult to predict what the balance is going to be in an intact tumor microenvironment in the patient.
Yeah. Yeah, and I guess you're trying to bring some of these therapies to the clinic. That is some of the big questions. You need to have some data in order to be able to test this in people. So how do we go about? We have this ideas from in vitro. We have mouse models. What are the steps that are becoming necessary to get to the point in which you can actually test this in a patient? You can actually test a two-pronged approach in a clinical trial.
Yeah, I think that depends on whether we want to target the tumor compartment or the T cell compartment. For the tumor cells, you need to take a pharmacologic approach. You need to develop a drug with systemic treatment, and hopefully this drug will reach most of the cancer cells in this context, for example, to sensitize them to the immune system. I think for targeting T cells, we now have an even more exciting opportunity. Of course, we have many cancer drugs, and the ones that made it to the clinic, many of them show robust responses in a single agent setting, probably in most cases not leading to a cure. But the downside of that is that you need a drug development program to develop the drug, and that can take 5 to 10 years. But the T cells, thanks to these function-based screens, which we and others have not only done in the cancer cells, but out in the T cells to make them fitter, to avoid or reduce the dysfunction, we now have a much more exciting opportunity, and that is genetic manipulation, right? So thanks to all of these screens and other approaches, we have now also for the T cells a very good understanding of the immune receptors, the key determinants of dysfunction, of exhaustion, of fitness, of T cell infiltration into the tumors, etc. And I think this is a very exciting development, because this unlocks a space for future cell therapies where we can take a genetic approach. So we can reprogram T cells with new antigen recognition receptors like CARs or TCRs. And taking advantage of all these genomic screens, we can do multiple manipulations. We can make knockouts, or we can activate genes. And we can do this simultaneously. And that allows us to boost T cell killing, to resist exhaustion, to neutralize immune checkpoints, avoid effects of immune suppressive ligands, et cetera, et cetera. And in the most recent developments, I think that it gets even more exciting, where people like Roy Cobo, Morrison, they also include control layers with logic gating and end gates, and that allows very precise activation of the T cells in the tumor microenvironment only in the context of specific cues that are desired. And you can include kill switches, and again, everything can be done by genetics, and that makes it, well, within immediate reach. And there's only one big disadvantage, and that's the price tag. So for regular CAR T therapy, it's about $500,000 per patient. But also there's light at the end of the tunnel with again an exciting development, which is in vivo manipulation, which I think is really the next major advancement for T cell therapy. So instead of harvesting the cells and engineer them ex vivo in a facility and then reinfusing them, now there are initiatives to reprogram the T cells directly inside the patient, and that is really exciting. And there's lots of activity in biotech space now towards that goal.
Yeah. If it works, it sounds a lot easier than having to take the cells out and then bring them back in. I think that we are reaching the end of our conversation today, and I know that. So we like to ask a little question at the end, talk more about more personal to our guests. And there is the question that we have for you, Daniel, is that if there is any advice, be it professional or personal or both, that you would like to share that helped you and you would like to share with the listeners, this is a good time to do that.
Yeah, that's interesting. So, so I like to answer that question in both ways, the personal one and the scientific one. So the personal one I got from my grandfather, who said that just show up do your best and be consistent. And if you stick to those basics, then good things tend to follow. And this is what I've tried to put into practice. And the scientific advice I got from the late David Livingston, where I did my postdoc in his department. So if I ran into him, he always asked me so hard things, and I started to describe my project. And then we said, stop. What's the question?
Right.
And that sounds like a cliche, but it's really not a cliche. I think it goes to the heart of the matter. And this is something that I try to pass on to my team members or elsewhere. If I get a seminar and I talk to the PhD students and the postdocs. And then, so when I ask them this, what's the question? And then they often start talking, and then they stop, and then they realize, oh, this is a bit more difficult than I had expected. And I think it's really important, if you can't summarize your work into simple question and then explain why that is important, maybe you should reconsider the project. So that's what I learned from him.
I think that's a very good advice. Sometimes it's easy to get caught on the everyday or the smaller things, and you're forgetting what
Right.
it's all supposed to be pointing towards, right?
Right. Yeah.
That's very good. Yeah. Focus matters. Execution is the other one. My grandpa and my father both gave the same advice. In the end, all that's measured is, did you execute?
That is true. That is true. And, well, I think that was very good advice. I think PhD students should take it into account. And anything else you would like to share with us, Daniel?
Yeah, maybe a little advice to the younger generations, the PhD postdocs. Everybody suffers from competition and pressure and frustration if an experiment fails or if a paper is not sent out for review or the grant is rejected. And many people I talk to, both at NKI but also outside, are considering careers outside of science, and that will be too bad because we really need those people. And we've all been there, and these are recurrent challenges. But I think it's also important to realize that we live in a time where we can use the most advanced technologies. Sometimes even like science fiction, what we do to see the RNA program of a single cell or the proteome of a single cell, and we can manipulate them in any way we want, and we really have a wonderful opportunity now to make a difference for the patients. I really think this is the golden age for science. So, scientists have I think the luxury to work in a treasury room and with a lot of space to make exciting discoveries. So my message to early career researchers is okay, we know all those challenges, but in spite of that, please try to enjoy this work. I think it's really special. It's a privilege to be a scientist and to make discoveries that change patients' lives.
For sure.
Well said.
That was great. Yeah. All right. So thank you so much. We had Daniel Peeper from the Netherlands Cancer Institute today in the show. Thank you for joining. It was a great conversation.
Thank you for having me, Jason and Brenda.
Thank you for being here.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. If you enjoyed the episode, feel free to share with a friend, colleague, or on social media. You can also reach out to us on X at Immunopodcast, on Blue Sky at immunologypodcast.com, or via email at info@immunologypodcast.com to have feedback or to suggest a guest. See you next time.